bed and what happens there. Okay, so before we get into that, let's talk a little bit about the anatomy that we're discussing. Okay, so at the end of the arterial system, right, we end up with our arterioles, the smallest vessels that still have a continuous coat of smooth muscle. All right, in other words, they are the smallest of the truly contractile vessels. Coming away from the arterioles, we have our capillary beds. Now, a capillary bed has a lot of little paths, traditional capillaries, connecting arteriole with venule. Okay, and we're going to talk a lot about what happens as blood goes from artery to vein. Guarding the entry of each capillary is a precapillary sphincter. You can see here, here, and here, which is really an extension of the arteriole. You can kind of think of it as almost part of the arteriole, these precapillary sphincters. The interesting thing about these is they're very effective vasoconstrictors. They can close that vessel off. In other words, the capillary bed can be open or closed because these precapillary sphincters are very strong and exert a fair bit of control, right? Now, we also see a couple of sort of more straight paths between artery and vein. What that is intended to convey is we do have bypass routes for blood to go from artery to vein without passing through a capillary bed. There are reasons why we would want that. Remember that what happens at the capillary beds is going to affect things like venous return coming back to the heart, right? So we have two kinds of bypass paths. The classic arteriovenous bypass, which is basically a largish capillary that directly connects arteriole and venule. And then we have something called the met arteriole. The met arteriole has a lot of control to it. So it can open or it can close. The arteriovenous bypass, you'll see, doesn't really have any sphincters along the way, right? So it's a little bit always open, where the met arteriole is variable, okay? So our precapillary sphincters exert the finest control over capillary flow. By opening and closing these sphincters, we control how much blood is in the capillaries and how rapidly it is flowing. It's the precapillary sphincters that are most affected by our local control factors that we've been talking about: pH, oxygen, CO2, temperature. All right, so the site of change of those variables is typically at the precapillary sphincters. The met arteriole also has responds to those same vessels. And while the met arteriole does create an artery-venous bypass, you'll notice that there are capillary connections, right? So the met arteriole isn't just about moving blood from artery to vein. It's actually about opening up new paths to fill the capillaries with, right? Because it's essentially like an arteriole that's been kind of turned on its side and is between the arterial and venous system. All right, so the precapillary sphincters and the met arterioles, they contract and relax a lot, all right? They're very sensitive to the local surroundings. So they create what's called vasomotion. If you were to study an individual capillary bed very carefully, you would see that blood flows and then stops, and then flows and then stops. Not like the heart is pumping, okay, not systole, diastole, but instead it's when the oxygen gets low and the CO2 gets high, these open, allow some blood to go through. That corrects that, so then it closes again, right? If the tissue's busy, same thing's going to happen. So vasomotion is the precapillary sphincters responding to the busyness of the tissue and causing more blood flow and less blood flow according to that tissue's needs. So the let's see, signs of tissue activity increase the amount of time that blood flows, right? So these are opening and closing. When a tissue is busy, they're open more often than they're closed, right? So overall, you get more blood flow through that vessel. Remember, these are very simple structures. Like the precapillary sphincters, there's not a lot of intelligence there, right? It's just smooth muscle wrapped around a vessel. So it can contract or it can relax. It can't like dial it in, all right? So instead of dialing in, we contract or relax more often, and that increases the amount of time that blood is flowing through those areas. All right, so if we zoom in even more and look at the cellular and molecular structures of the capillary bed, particularly the capillary itself, we can see capillary walls are incredibly thin. Some of the thinnest cells in the body are the endothelial cells. They are modified squamous epithelium, only they're not stratified, right? You know, like you're used to seeing stacks of squamous epithelium. These are squamous epithelium in a single layer, right? The endothelial cells are. So this is a cell, this is a cell, this is a cell, and you're literally looking at it from the side, like most of the cell is out here in our picture. There are a number of ways for materials to pass through the wall of the capillary. And that's important because that's where the business is getting done, right? So we need to have our nutrients leaving and our waste products coming in. So the barrier between inside the capillary and outside the capillary is this single cell thickness. Now, in between the cells, there is a cleft called the intercellular cleft, and that allows some molecules to pass through, okay? We also have vesicular system called the caveolae, right? And that's what this is attempting to show you. In the structures here, I'm not really interested in you knowing the structures, but caveolin is the protein that creates these little vesicles. And these vesicles are constantly moving materials from inside the capillary outside and outside to inside, all right? You can get a bunch of these caveolae that if they form in a line, you can get a kind of vesicular channel, a sort of pathway through the cell. But that's relatively uncommon. Usually we have these little sort of indentations where pinocytosis occurs and materials are exchanged. Okay, so how do things get from inside to outside and vice versa? One path is through the intercellular clefts, okay? Another path is through the cell membrane, right? And then a third path is, well, it still is across the cell membrane, but receptor mediated, right? Where you actually have endothelial cells that are binding ligands and bringing them through. Most of the flow of materials at the capillary bed is the first two. It's through the capillary membrane or through the intercellular clefts. Now, not all capillaries have the same structure. So you might remember from your previous anatomy and physiology experience that the brain has what's called the blood-brain barrier, right? Where it's harder for things in the blood to get out into the brain. Well, one of the reasons for that specialized barrier structure is the intercellular clefts of the capillaries in the brain are bound together by desmosomes, which means that materials in the brain can't pass between the cells. They can only pass through the cells. So it means that anything that goes from blood to brain or brain to blood has to pass through a capillary through a capillary cell itself in order to occur. It makes a very rigid filtration system, right? There's no way to go around the cell. You have to go through the cell. And the cell is the guard that either prevents or allows things to pass through. Sort of the opposite state is in the liver. In the liver, the capillaries of the liver sinusoids have very big gaps between them, it is very easy for materials to get from the blood into the liver and from the liver back into the blood. This makes perfect sense because the liver is our metabolic powerhouse, right? So we want it to be easy for the liver to process things that are passing through the blood. The other thing that this allows is when the clefts are wide enough, white cells can squeeze through, right? So we have in the liver as well as in the bone marrow, we have these wide intercellular clefts so that cells can come into the blood and leave the blood as necessary. In GI capillaries, the clefts are wide for a different reason, and that's to allow nutrient absorption. We're going to talk about that in our GI tract discussion. And then the glomerular capillaries in the kidney, they have sort of specialized holes in their membrane called fenestrae, which are it's a fancy name for window. But the capillaries of the kidney it's almost like a colander, like you would use to drain spaghetti, right? So there's lots of little holes that allow fluid to pass, but not proteins to pass. So small molecules can pass through, but not large molecules, and that's part of the filtration system. Well,